Bio 181, Chapter 13, Human Inheritance. Key concept here is that in human females, the inactivated X chromosome varies from cell to cell. So the sex chromosomes, you have a pair, if you're female, of the X chromosome. So you have two of those, but in any given cell, only one of them is going to be active and the other one is going to be condensed. So it's going to be small, it's not going to be actively read. So you've got a zygote. The zygote is formed, it's the single cell that forms as a result of the fusion of the sperm, which again in humans is bringing in 23 chromosomes, the egg, which has 23 chromosomes, 23 chromosomes. All right, I'm just going to do that, 23 chromosomes. So then in the zygote, right, we would have 23 plus 23, which would be our 46, which is the amount of chromosomes in a human. For the sex chromosomes, you have, if you're a female, you got that 23rd pair, that's these homologous X chromosomes. And so you have random inactivation. So this is showing here that by the different colors. And when the cell divides through, again, this is going to be through mitosis, then in the nucleus, only one of the X chromosomes is going to actively be read. So in this cell, the genes are going to be from this one at random, and in this cell, the genes are from this one at random. So these will have different alleles, okay? Different alleles, not always, right? If it's homozygous, but if they're heterozygous, they can have different alleles. So then you're going to have an assortment of cells that may be expressing different alleles and function slightly differently. So an illustration of this very clearly is in a cat. So these calico cats are always used as the go-to example because it's very well illustrated. You can look at the colors on the coat. And so the different regions of the cat correspond to cells that have been activated. One X chromosome has been activated, whereas a different color, in this case the black, a different X chromosome has been activated. And one of them has an allele for black fur and one of them has an allele for orange fur. So it's super easy to view. Another concept, so I'm shifting gears a little bit here, is the idea of mitochondrial genes, and they are inherited only through the mother. So this is called maternal inheritance. So organelles that are passed down, if you have a sperm coming in, the sperm's actually really little, and it basically has, some DNA in here, but not much else, right? Here's a big egg cell, right? So then the egg cell, it has some DNA, but it also has a lot of other things in it. It's got mitochondria in it. If it's a plant, it's got chloroplasts, right? So it's got all of the other organelles. So the mitochondria has its own DNA. So there's a small circular chromosome of DNA in the mitochondria. And that's going to be inherited only through the mother. So when the sperm and the egg fuse down here, right? So the sperm fuses here, it basically just drops off the DNA in here. So you've got twice the component of DNA, but then everything else in this newly formed zygote is going to be from the mom. So you can trace genetic lineages this way. So mitochondrial inheritance has really been useful for tracing the evolution of human traits over time. And this is MT, here is just the abbreviation for the mitochondrial DNA. And you can see that it's passed down from the grandmother to the mother to the offspring, which would be the daughter and the son here. Okay. And then the last concept for this video, there's this kind of a mishmash here, is epigenetic inheritance. And this is a really fun, evolving field where there's tons of research going on, lots of interest. in terms of inheritance patterns that can change gene function based on the environment and things that are not coded for in the DNA. So over here, you can see there can be effects from the environment that can change how the DNA is packaged, or some extra chemicals that are added to the DNA. And that's going to regulate which genes are expressed and which genes are repressed. And that can change the phenotype or the physical expression. So previously, years ago, it was thought that, okay, genes are always just determined by the DNA. And so if you know the DNA sequence, then you're going to know exactly what the phenotype is going to be. But in fact, there is this interaction with the environment, which we're still learning about the extent to that. So epigenetic traits, defining that, these are stable heritable traits. So this is passed down from parent to offspring, and it cannot be explained by changes in the DNA sequence. So it's not a mutation in the DNA. So what does that look like? Well, you've got your chromosome here. This is and you have a sequence of genes. If we were to zoom in on this chromosome, you would find that the DNA, right, is actually it's really long. And so it's wrapped around these proteins called histones that help package it. The DNA is negatively charged. These histone proteins are positively charged, and it helps condense the DNA into a small package. These are histone proteins. Okay. So chromosomes are made up of DNA and proteins that help with the packaging. And so sometimes these proteins are actually modified chemically. So you can add like methyl groups to them or other groups, and that can change how easily it is to get at this DNA that's next to it and read it off. Similarly, if you actually look at the DNA itself, so let me go back here. If we were to zoom in closer on this DNA, whoops, just keep it blue. So keep it here. So this DNA is a double helix, which we'll learn more about soon. So it's got a sequence of nucleotides, which are the monomers of nucleic acids. And on here, these sequences of DNA can actually be methylated too, or you can add other small chemical compounds, or this is a functional group that we learned, methyl, if you remember that, METHYL was CH3. So these are often added on, and they've been shown to activate or repress genes. So methylation, I just mentioned, this is shown up here, cytosine. This is a base that's in a nucleotide. We'll talk about this later, but you have A, T, C's, and G's. And cytosine is often methylated, and that's shown here. So we're adding on this extra bond, CH3, this bond here. We're adding on a carbon and a few hydrogens, and that again can promote or repress the expression of genes. We also learned about phosphorylation previously, and phosphorylation of the histones is also associated with epigenetics. Here, serine is an example. A serine kinase, remember kinase, ASE on the end means it's an enzyme. And in this case, it's adding a phosphate group. So it's taking adenosine triphosphate. The phosphate group is going to be transferred. Oh, it's actually up here. So that's the phosphate group. That's the P, right? So then the adenosine triphosphate turns into the adenosine diphosphate, and the phosphate group goes over here. So that action also is associated with epigenetics. All right, that's it for this video.